Today we'll be talking about prokaryotic replication. To do that first, we should get to know a prokaryote. Here's Lucy the prokaryote. In order for Lucy to successfully replicate herself, she must go through the replication fork with little to no air. First, let's look at some of the major chemical and physical aspects of DNA. It has a right-hand coil, a deoxyribose sugar, a phosphate backbone, and 4 nitrogenous bases: adenine, thymine, guanine, and cytosine. Phosphodiester bonds are formed between complementary base pairs. Now, let's look at some of the specific steps and parts concerning the prokaryotic replication fork. One of the first things that needs to happen is that enzyme helicase needs to bind to the template DNA strand at the origin of replication. The more phosphodiester bonds helicase breaks, the more coiled the DNA becomes ahead of the replication fork. This is where DNA gyrase comes in. DNA gyrase is a form of topoisomerase that breaks the phosphate backbone of DNA for a temporary period of time to relieve coilage throughout the remaining process. After this, single-strand proteins enter and bind to each of the 2 new single strands to prevent the double helix from reforming. Then, RNA primase places an RNA primer somewhere along the strand. DNA polymerase III finds this primer and then uses it to begin synthesizing a new strand of DNA. DNA is anti-parallel, meaning the 2 strands show opposite orientation to one another. Because of this anti-parallel nature, each strand has its own set of unique properties and methods of replication. One strand is called the leading strand. This is the strand that follows the 5' to 3' orientation. This strand moves toward the direction of the fork, allowing it to synthesize continuously until termination. The other strand is called the lagging strand. This strand has the orientation 3' to 5'. This direction is moving away from the replication fork, which induces stress. This stress results in small fragments of DNA being produced called Okazaki fragments. It is called the lagging strand due to the fact that it lags behind the leading strand when it comes to efficient replication. The gaps between the Okazaki fragments can't remain there if Lucy wants functioning offspring, so DNA ligase comes in and seals them all together. Meanwhile, DNA polymerase I replaces the RNA primer found earlier in the process with DNA. If Lucy can make it through all these steps with no error, she will become a very genetically successful prokaryote.